So next we'll hear from one of our 2018 Next Generation leaders, Maria Tosches. Maria is an assistant professor at Columbia University in the Department of Biological Sciences. Her research is focused on the evolution of neuronal cell types and neural circuits. She received her PhD from the European Molecular Biology Laboratory in the lab of Detlef Arendt, where she studied melatonin signaling in neural circuits of circadian states in a marine worm. As a postdoc at the Max Planck Institute for Brain Research with Gilles Laurent, she used single-cell transcriptomics to study the evolutionary history of cortical, glutamatergic, and GABAergic neurons in turtles and lizards, so very different species from what we're used to studying here. Today she'll give a talk titled The Evolutionary Origins of Cortical Cell Types. So please join me in welcoming Maria. Thank you.
Thank you. It is a great honor to be here. And I'm looking forward to the rest of the showcase to learn more about the new things happening at the Institute. And I have to say that since my PhD times when I was working on annelid worms, the data and the resources generated here have been crucial for my research. Because my research is comparative. I'm interested in learning about brain evolution from comparing different species. And the knowledge base built here for the mouse brain and lately the human brain has been fundamental for these comparisons. So first of all, why evolution? Like all of us, I'm also interested in understanding the brain at a fundamental, deep level. And when we think about the brain, we usually think about, we like to compare the brain to computers. And we think about how the brain is built to solve these very complicated problems and sort of run algorithms. But this analogy is missing something. And what's missing is the fact that the brain was not engineered to be the optimal machine to run certain algorithms, but is the product of an evolutionary process. And this is when the complexity of biology kicks in. Evolution of the brain started with the appearance of the first neurons more than 700 million years ago, and then the appearance of the first nervous systems. And there's been no purpose in this evolutionary process. The human brain is not the end goal. Actually, what makes biological systems successful is their capability to continuously evolve, or evolvability. So there are mechanisms built in our brains that ensure that not only they are flexible enough to adapt to different conditions, but they're also prone to change and to evolve over time. And so for all these reasons, I think it is going to be very important to have an evolutionary perspective on the brain, just because this will help us to understand the fundamental principles of brain organization and function. Just because the brain was, as I said, not a product of engineering and optimal design. So how do we go about this? Of course, we cannot go back in time and look at the brains of our ancestors. But what we can do is to compare brains across species. And I'm interested in these large-scale comparisons, going beyond the human mouse and actually looking at non-mammalian vertebrates. Before I talk to you about this, well, I want to mention what we can learn from these comparative studies. Well, what we do is essentially to look at similarities and differences. And similarities are the result of two different processes. They can arise from homology, which means when from descendants from a common origin. So two characters, for example, cell types, are homologous if they existed already in the common ancestor. But similarities can also result from convergent evolution, which means the two characters independently became similar, even though this character was not present in the common ancestor. And these two processes, homology and convergence, are giving us different information about the brain and brain evolution, because homologous characters are characters that have been maintained over millions of years. So presumably, the conservation of these characters reflects their importance for, in our case, for brain function. Convergence, instead, indicates that probably there are developmental functional constraints on nervous systems that push the evolution of similar features. for example, similar circuit architectures to deal with similar sensory environments. So when we do our comparative work, we are looking at these similarities and trying to understand whether they arise from homology or convergent evolution. And as I said, I'm interested in the vertebrate nervous system. In my postdoc, I worked on reptiles. I started my lab 2 months ago, and I'm now busy bringing in a new species to the lab. This is a salamander species that I will tell you about later in the talk. But the overall idea is to compare this species to the mammalian brain in order to reconstruct or to infer features that might have been present in the amniote ancestor and in the tetrapod ancestor. What is really striking when you look at vertebrate evolution is that it's a long history that started more than 500 million years ago. And nevertheless, the overall organization of the vertebrate brain is the same, meaning that we find the same regions. We find a telencephalon, a brainstem, or a cerebellum in each vertebrate species. So this is because these brain regions are set up very early in development when conserved signaling pathways and transcription factor cascades regionalize the developing brain. And what then gets interesting is that from this common plan, or from this common organization, differences are built in, which account for the species-specific differences. And this happens through the divergence of developmental programs that leads to the diversification of cell types, of neural circuits, and tissue architecture. And I must say that this divergence process is still not very well understood. And that's one of the topics of my research. These differences can be really dramatic. So I'm going to show you how the telencephalon of a turtle and how the telencephalon of a lizard look like. As you can see, at the first look, it's very difficult to recognize similarities with the mouse brain. Nevertheless, we can still define a dorsal telencephalon on the basis of gene expression data and anatomical data, which is also what we call pallium, and is distinct from the ventral telencephalon. And the dorsal telencephalon of these animals also has a cerebral cortex, even though it's much simpler and smaller than the mammalian cortex. So the cortex here is defined as this, as you can see, this sheet of cells that are arranged in layers. The difference with the mammalian cortex is that in the center of this cortical sheet, in mammals, there's been an expansion from a simple 3-layered cortex to a 6-layered cortex, the isocortex we are all familiar with. Whereas in reptiles, the cortical sheet has this 3-layered organization throughout its whole extent, from medial to lateral. And what these three layers means is essentially a central layer, layer 2, of glutamatergic neurons, and the 2 layers of axons and dendrites with GABAergic neurons interspersed throughout this cortical sheet. In the pallium, there's not just cortex, but there are also non-cortical structures or nuclear structures. In mammals, they comprise small nuclei, such as the claustrum and the nuclei of the amygdala. In reptiles, the nuclear structure goes under the name of DVR, which stands for dorsal ventricular ridge. And on the basis of anatomical data, it has been extremely difficult to infer anything in terms of evolutionary relationships between this DVR and parts of the mammalian brain. So there's a long history of literature where people fight over different camps and different theories about how these brains are related to each other. And I'm not going to summarize that for you. I just want to mention that these big conflicts arise from the fact that people have been focusing on the overall anatomy and architecture of these brains. And because the differences are so large, it's difficult to pick up commonalities. So the strategy that we've been using instead is to zoom a little bit in and look at the building blocks of these pallial regions, which means to look at cell types. And that's what I'm going to tell you in the rest of my talk. We use single-cell transcriptomics to figure out what are the reptilian glutamatergic and GABAergic neurons in cortex, and what are their relationships to mammalian neurons. We also studied the anatomical organization of these reptilian brains in light of the single-cell data, and this provided some insights on the evolution of cortical layering and on the nature of the DVR. So we collected data from this turtle species, which is an American species, Trachemys scripta, and this first data set was generated with drop-seq and from the pallium of these turtles. And right now we're working on a second data set. We finished with the acquisition and we are analyzing the data. This second data set comes from an Australian lizard, the bearded dragon, and it's a data set collected with 10x genomics where we are actually exploring the entire brain. So I will mention a few of the key results from the lizard data as well. And I have to say that the most time-consuming part of these projects has been not collecting the single-cell data, but relating them to the anatomy. Because we, as you can imagine, we have no anatomical atlas for reptiles. So we have invested a significant amount of time in generating small-scale atlases, gene expression atlases, where we cloned and we performed in situ hybridizations on over 100 genes for each of the two species. And this was a large collaborative effort within my postdoc lab, which involved students, other postdocs, and technicians. And by using this data in conjunction with the single-cell data, we've been able to map our cell type clusters on the anatomy, because we used the single-cell data as a guide to select the most specific markers and then do the in situs only for those cluster-specific genes. For the rest of the presentation, I want to walk you through two main take-home messages that come from the comparative analysis of this data. And I think these are points that are true for cortex and pallium, but might be also more generalizable in terms of brain evolution. So the first point is, what we see is that the elements of cortical circuits are, in terms of the evolutionary history, they are mosaics of new and ancient parts, meaning that cells that come together and are functionally together in a circuit can have very distinct evolutionary histories. And that was the first surprise for us. So this, as I said, we found this for cortex, but we think that this conclusion might extend for the rest of the brain. Before I go into the data, I just want to say a little bit about how we think about these large-scale comparisons of cells, of cell types. First of all, the underlying idea is that cellular identity is established by a set of transcription factors, which we call terminal selector genes, that are the transcription factors that activate the cell type-specific effector genes, and that they truly identify as a cell type, as a distinct type from other cells. So in this hypothetical scenario, what I'm showing you are two cell types that are similar to each other, that they express the same terminal selector genes, and they're found in two different species. So the inference we make in this case is that the ancestor of these two species had the same cell type, presumably with the same terminal selectors. And so this is a case that we call cell type homology. Of course, because we are comparing species across these very large distances, the fact that you have similar sets of same sets of terminal selectors does not necessarily imply that the transcriptomes are identical. And as we have seen from your mouse to human comparison, even there are differences at the level of individual effector genes. These differences are actually even more substantial in these large-scale comparisons. a different case arises when cellular diversity is expanded through a process that we call cell type diversification. The idea behind this is that what happens in brain evolution is that these transcription factor codes can change, and for example, by adding new transcription factors to the set of those transcription factors that define cellular identity, or removing some of these. And as a result of this change, you have an expansion of cellular diversity that starts from a cell type that was present in the ancestor. So this cell type diversification is one of the many mechanisms you can imagine evolution uses to generate new cellular diversity. And we have to, and this is, let's say, our theoretical frameworks to think about cell type diversification and to recognize cell type diversification in the sequencing data. So what did our data look like? This is your GABAergic data set from the 2016 paper, which shows where I selected some of the key markers, including transcription factors that are shown in red, that define these main classes of GABAergic neurons in mouse cortex. And what was a big surprise for us was to see that this exact same markers are sufficient to define 3 classes of GABAergic neurons also in the turtle cortex, suggesting that these CG-derived serotonin receptor neurons, somatostatin and PV neurons also exist in turtles. And I have to say, few of the key markers, such as parvalbumin itself, are not expressed in turtles, but overall, these combinatorial codes, which include the terminal selector transcription factors, are the same. What was even more surprising was to see in our new lizard data set that the similarities go deeper. They're not limited to cell classes, but they go down to the individual cell types. And we achieved this by expanding our data set with the new lizard data, and then using canonical correlation analysis as a basis for integrating these multimodal data sets across species. And what you see in this UMAP in the center is the joint dimensionality reduction of our new lizard data with our, with mouse data publicly available. And you see that these data sets align very beautifully. This leads us to the identification of homologies or correspondences between mouse and lizard GABAergic types to an extent that we could not predict with our turtle data sets, just because our previous data set was too limited with a few cells. There are examples of types from mouse that don't have a lizard counterpart, as you see, for example, down here. But overall, we found that the major types of GABAergic neurons that exist in mouse also have a counterpart in lizards. So now, going to glutamatergic neurons, we've repeated the same exercise. We've taken the main transcription factors and key marker genes from mouse data, and we've used the same genes to look at our turtle glutamatergic neurons. And here the story is completely different. As you can see, these genes now do not help us at all to classify turtle glutamatergic types. It looks like these genes are either expressed everywhere or not expressed at all in the turtle dorsal cortex. So there are 2 main takeaway messages here. One is that overall the transcription factors are there. The missing genes are more of the effector kind of genes. But the transcription factors are all found in turtles. The difference is that they're not found in the same combinatorial codes that exist in mammals. But they are found in completely different combinatorial codes, and usually they are more co-expressed together rather than segregated and mutually exclusive. So these results struck us a little bit because at first it seemed that, okay, turtle and mouse glutamatergic neurons, can't be compared at all. What do we do about it? And so what we did was to do a more shallow comparison and look at correlations of these transcriptomes. And again, as a surprise, we found that the turtle glutamatergic types fall into two groups. The ones that have higher correlations to cortical upper layers from mouse, and the ones that have correlations to deep layers. And then so at that point, we went on and looked at marker genes to understand where these upper layer-like and deeper layer-like cells from turtles are localized in the turtle cortex. And we saw that indeed this layer 2, where all the glutamatergic neurons from turtle are found, is subdivided in two layers that can be recognized only by molecular markers. They're not anatomically distinct. And these two layers correspond to these upper layer-like and deeper layer-like cells. What's even more intriguing is that their topology is inverted. So you find deep layer-like neurons on the surface, and the upper layer-like neurons deeper, closer to the ventricular side. And that's because it was already known in the field in non-mammals, corticogenesis does not have the inside-out organization, but it has an outside-in organization. So early-born neurons are found more superficially, and then late-born neurons are just accumulating once they're born on the ventricular side. So this topology, this organization is actually a reflection of the developmental process. And what we think is that these very shallow similarities might be an indication of a cell type diversification history. So to sum this up, we think that GABAergic types are homologous across reptiles and mammals. And in contrast, glutamatergic types in mammals might have evolved through this diversification process with a segregation of gene regulatory modules that, and elaboration of these gene regulatory networks that define cellular identity. And with this diversification model, we can also explain how this new morphology with the six-layered organization might have appeared. So that was my first point. My second general point is that what we find ancient cell types, such as these GABAergic neurons, but there's more of these ancient cell types, very often hidden in diverging tissue architectures. So now I'm going to take you outside of the isocortex-neocortex story and look more generally into pallium. And there's two places where we find these very deeply conserved cell types in diverging tissue architectures. And one is the hippocampus. The hippocampus is the medial part of the cortical sheath. And in mammals, it is folded and embedded, pushed deep into the brain. But in reptiles, it remains contiguous to the rest of the cortical sheath. And it was very long debated in the field whether there is a dentate gyrus and whether there are CA fields in a reptilian hippocampus. And what we were able to see using marker genes and single-cell transcriptomics data is that also in reptiles, the hippocampus is subdivided into a more medial zone, which is the dentate gyrus, and a more lateral zone, which is the CA field, that is further subdivided into regions that seem homologous to CA3 and CA1. There's also a second example of ancient cell types that are embedded in different morphologies. And this was even a bigger surprise for us, totally unexpected, and it's the example of the claustrum. So the claustrum is, as you well know very well, is deep into the brain, squeezed under the insular cortex. And there is nothing that morphologically looks like that in a reptilian brain. But what we've been able to see with single-cell data is that there are parts of this big DVR mass in reptiles that are homologous to the claustrum. And because this was previously unrecognized, these claustrum homologs have been called with different names. In turtles, this is the so-called pallial thickening. And in lizard, this region was not even recognized as a separate anatomical region, but it's something we discovered just out of the transcriptomics data. So how did we get there? This came from our new data set from lizards, where we used marker genes that were specific of these clusters to map these clusters on the anatomy. And we found that this very distinct cluster of glutamatergic neurons maps to this region of the lizard DVR. And when we started to look at the marker genes typical of these two clusters, we realized that many of these marker genes are typical claustrum markers, such as GNG2 or RGS12. So that was suggestive, and that pushed us to do a more comprehensive comparative analysis, where we have taken data from mouse encompassing the entire pallium, and we have done a joint dimensionality reduction with our new lizard data. And in this plot here, I'm going to show you the percentage of lizard cells that map on mouse clusters. And you see some mapping at the level of hippocampus, as expected, not so much at the level of cortical, isocortical clusters, again, as expected. And then this very strong match between our lizard claustrum cells and the mouse claustrum cells, which was very exciting. What makes these lizard cells so distinct and so special? Among other things, it's a whole suite of receptors for noradrenaline, acetylcholine, dopamine, and serotonin. These are the only glutamatergic neurons in the reptilian pallium that receive information from all these neuromodulators simultaneously. And this prompted us to look at connectivity. So this was work from Xin-Chi Li and Lawrence Fenk in the lab, where they used AAVs to map connections between a claustrum and other brain regions. And as predicted by the transcriptomics, they found that all these midbrain and brainstem nuclei that provide information on brain state are projecting to the reptilian claustrum, and that the reptilian claustrum is reciprocally connected to cortical areas in lizards, which was really rewarding. What is this claustrum doing? The fact that this is such a massive region in the lizard brain makes it accessible for experimentation. And Hiro Norimoto and Lawrence Fenk went on to look at the function of this region by doing electrophysiology in vivo and ex vivo. And to make a very long story short, what they found is that this is the source of a very specific electrophysiological signature that appears during sleep, during the slow wave phase of sleep. This signature is called sharp wave ripple, where you have a deflection of the local field potential with action potential on the trough of the sharp wave. And when you ablate the claustrum, these sharp wave ripples are completely gone, indicating that the lizard claustrum is essential for the generation of the ripple and the propagation of the ripples to the rest of the telencephalon. So suggesting a role for this claustrum in the sleep. To summarize, I showed you that single-cell transcriptomics is a fantastic tool to generate quickly cell-type atlases in non-conventional models, and this can really bring us to a new level of comparative analysis across species. Going to cortex, our main result is that cortical circuits are mosaics of cell types with completely different histories. Some are very ancient and some are totally new. And that, in general, we find these very ancient types, such as claustrum cells, hidden in diverging morphologies. As I mentioned earlier, in my new lab, I'm going to focus on salamanders and take on some of the questions that follow up from these studies. The two main branches of my group will be investigating mechanisms of neuronal diversification by looking at gene regulatory networks and lineages throughout development in order to understand how this neuronal diversification process might happen in evolution. But also very interested in understanding the consequences of this coevolution of neuron types and the fact that neurons with diverging evolutionary histories can come together to form, to work together and to form circuits. How is this constraining neuronal evolution and vice versa, circuit evolution? And I'm going to do this with the Spanish-ribbed newt, which is an amphibian. So it's interesting for understanding the transition from aquatic to a terrestrial lifestyle. In the vertebrate history, it's useful for inferring, again, what happened at the divergence between amniotes. It has a much simpler brain than the reptilian brain, one of the smallest brains of all vertebrates, which should be very useful for applying large-scale systems approaches, and a series of tools and resources such as transgenesis, which is impossible in reptiles, or nearly impossible, and regeneration, genome and transcriptomics available. And with this, I would like to thank my postdoc advisor that supported all the reptilian work, Gilles Laurent, for his vision and his encouragement. I want to thank my colleagues in the Laurent lab that really joined me as a big team to first tackle the turtle pallium, and now with Tatiana and David, the big challenge of working on the entire brain. It's still a relatively small team. And then I want to thank the EMBL Gene Core that supported us at the beginning, especially with getting all these single-cell technologies up and running, and my collaborators at Karolinska for support on the new work on salamanders. And thank you for your attention.
Okay, we have time for a few questions.
Great talk. I wonder if you can, so the claustrum sharp wave ripple result is really striking and phenomenal, I guess. Have you looked into whether the sharp wave ripples are also occurring in the hippocampal analog that you also beautifully showed, and whether eliminating the hippocampus eliminates the sharp wave ripples as well? I mean, it seems like maybe to play devil's advocate, disrupting the circuit in such a large way as removing a big brain region might have disrupted sharp wave ripples, not in a specific way, but in a non-specific way.
Okay, so this is ongoing work in my postdoc lab, and I have to say, the hippocampus, it's of course the place to go. It's technically very challenging because it's very thin sheet and probes are not very stable, but people are working on it. But yes.
I have a relatively general question. We like to talk about evolution, thinking about how systems are pushed in different directions, cell diversification happens. But at the same time, we understand the principles of evolution, which are going to be with selection. Is there anybody trying to link these elements, which are going to tell you how this cell type diversification is going to end up bringing any type of evolutionary fitness?
In a way, this is touching on what I want to do at a very high level in my lab. I think that we have, with single-cell transcriptomics, we have these great tools for comparing molecular fingerprints of cells across species, but we are missing exactly what you're saying. We're missing an understanding of what are the evolutionary pressures that are shaping the evolution of these cell types. And I think we are going to miss that unless we go into studying neural circuits, because a neuron by itself is not doing much, but neurons are functional within the context of a circuit. So one of the part of the things I want to do is to link these evolution of cell types to the evolution of circuits, and study, for example, whether these conserved cell types participate in conserved circuit motifs, or how is the evolution of gene modules within these cell types related to, for example, new circuit functions and circuit motifs. So yes, this is one of the future goals.
Thanks. I have a similar question. I think that based on the way you presented the results, which are really interesting, it seems like thinking about the fitness that evolution is serving, you could think about it either as species evolving to serve some like fitness of the species, like survival, or neurons evolving to fulfill some fitness of like a neural circuit. Do you think it matters which way you think about it, and do you think about it one way or the other?
I'm not necessarily set. As I mentioned earlier, I think that understanding the relationship between evolution of neurons and evolution of circuits is going to be a critical point. After all, what serves best the organism is the capacity of adapting and surviving its environment. For that, I think we're still very early in understanding how the brain is operating at all these different time scales. It's not just about the fast circuit operations, but it's also about these very long, slow time scales that I think might play a role into the capacity of a brain to adapt and to perform well in a certain environment. So I think it's still relatively early to say what's the best level to look at. My bet is maybe looking at the interaction between, or relationship between cell types and circuits, and then we will see from there.
Okay, well, let's thank Maria again for a fantastic talk.